Welcome back, children. Last class we saw the basic introduction about the genetics, that is principles of inheritance and variation. Now we go to see some basics before performing a cross that you all must know. The first basic you all must know is P1. Commonly we use a term called P1. What does P1 signifies in a cross? Most of them do not know. They simply put P1 and P2, F1, F2. What does P1 stands for? That is very important. The P1 stands for parents of F1 generation. Okay, fine. Parents of F1 generation. What should be the character of the parent? They must be pure breeding. The other name for pure breeding is homozygous. I'll tell you what is homozygous. They must be homozygous, where there are two types of homozygous: homozygous dominant and homozygous recessive. Therefore, the first unique character that we have to see is P1. P1 stands for parents of F1 generation. The character they must possess is they must be a pure breeding, and scientifically we call them as homozygous. There are two types of homozygous we have, that is homozygous dominant, homozygous recessive. To understand that, capital T is called as homozygous dominant, small t and small t is called as homozygous recessive. Okay, fine. Next is F1, a term came here. What is F1? F1 is nothing but the progeny obtained by crossing the parents, or that is the P1. So you can see here we call this first filial generation. So how this first filial generation is obtained? It is obtained by crossing the parents of the first generation. We are obtaining the F1. So what is P2? That is the second technical term that you must know. It is nothing but parents of second filial, that is F2 generation. So what is the condition of this parent means? The parents P2 will be always heterozygous. So that is nothing but a hybrid, common term. The hybrid is nothing but an heterozygous. So heterozygous means what? Capital T and small t. This is a heterozygous. How this heterozygous means parents are, so these parents are obtained, how their parents are obtained means the F1 itself is the parent of the F2 generation, P2 generation. So what are these generation means? F1 into F1, because F1 progeny is always a heterozygous. Next is F2 generation. What does F2 generation means? It means second filial generation. It means second filial generation. So we have the P1, F1, P2, F2. These are the common things that you must know. These are the common things you must know. Next, we are going to talk something about Punnett Square. So what is Punnett Square? It is a probability square to find the zygotes of the cross. In other way, you can see this how we have to perform a Punnett Square. You have to put a square and you have to put a double line. And here we are differentiating between male and female, and put a line here, and here you have to put the gametes. For example, X, Y, X, X. That means here I have taken the female gametes, here I have taken the male gametes. Crossing these two, the probability of the zygotes are XX, XY, XX, XY, 50-50% chance. So Punnett square we have to always perform only for the F2 generation. We will not be performing it for the F1 generation. F1 generation always resemble any one of the parent and they are heterozygous. Now the question we have here is homozygous. Homozygous means what? Homozygous means true breeding plants. That means we have two types. Already we have discussed about that. We have dominant. Next we have recessive. Dominant, what do you mean by dominant? Dominant traits are expressed. Dominant traits are expressed. It is easily expressed in heterozygous. But when you talk about recessive, it is suppressed in heterozygous. So when it can be revealed means the recessive trait will be expressed only in homozygous recessive area only. Now very important thing that we going to take into consideration is genotype. What is genotype? Genotype is the genetic constituent of an organism. Genotype is nothing but it is the genetic constituent of an organism. For example, based on your grade 10 knowledge, we have three types of genotype in monohybrid cross: capital T, capital T, capital T, small t in a heterozygous, and small t, homozygous recessive. For this, we will be putting something called phenotype. What do you mean by phenotype? Means it is the external observable character. It is the external observable character. And the term genotype and phenotype for neat examination, it was coined by Johansson. So capital T means it is a genetic constituent exhibiting the tall character and is a homozygous dominant. Capital T, small t is in heterozygous and dominant traits are always expressed in heterozygous. Therefore, this plant is also tall. Genotypically, it is a heterozygous. Phenotypically, they are expressing the trait called as tall. And small t is called as the dwarf. Small is called as the. Yeah, children. Now, a very important doubt which most of the students have is: so what is gene? Okay, what is DNA? Followed by what is gene? What is an allele? Most of the students, they have this doubt. What is DNA? In addition, what is chromosome also? To understand that, I'll tell you a simple basics. It is a nuclear membrane. And you know it is a discontinuous membrane because they have the nuclear core. In this, you have some thread-like structures. What is the thread-like structure? This thread-like structure is called as chromatin. This chromatin, they will condense and they will form one X-shaped structure, and this X-shaped structure is called as chromosome. So chromatin condense to form chromosome. What is chromosome made up of? Chromosome is made up of DNA plus proteins. So how the structure resembles means we have a small protein, children. This is called a positively charged protein called histone proteins, and the charge of DNA is negative. Why the charge of DNA is negative? Because of phosphoric acid, the charge of DNA is negative. So the negative charged DNA wraps around this positive charge structure like this. This concept is called as nucleosome. What is nucleosome made up of? It is made up of 200 base pair of DNA plus 8 types of histone proteins. What are they? We will be discussing about in second chapter of genetics, histone proteins. Therefore, what is a chromosome? Chromosome is made up of DNA and a protein. And next we will take into consideration from this we will take some thread-like structure like this. This thread-like structure is called as your DNA. The DNA, we have some nitrogenous bases. These are nothing but genes. In the genes only we have something called as allele. So the common thing, what is a gene? Gene is the unit of inheritance. People say gene is the unit of inheritance. Gene is the unit of inheritance, and commonly we call it as a segment of DNA. So gene is a small segment of DNA. In other ways, we can call them as the nitrogenous base are adenine, thymine, guanine, cytosine. These are the nitrogenous bases. These nitrogenous bases forms a type of concept called as codons. Some concept we will be learning about that, where they will be always in the form of three letters A, T, G, and like this A, G, C. Or in other ways, A will be replaced by U. Some concepts are there where the T is replaced by U in case of RNA. So about a concept we will be discussing here when it comes into account of DNA. So what is a gene? Gene is a small segment of DNA and it is a unit of inheritance. So the genes will come in multiple versions. So the multiple versions of gene is called as allele. For example, if I take a chromosome like this, capital T allele is here, small t allele is here. So allele is location of the genes controlling a particular character. Okay, children. Now we could discuss about Mendel and his experiment. Before starting an experiment, we must know what is the experimental material that he have taken for this experiment. The experimental material is nothing but a simple plant called garden pysaevum or garden pea plant called Pisum sativum. The board exam half mark will be reduced if genus and species are not separately underlined. Follow the rules of binomial nomenclature. Commonly we call it as garden pea plant. So the pea plant was taken for his experiment. How many years he have done his experiment means he did work with this plant for 7 consecutive years from 1856. Even we are not born. It's in the 18th century the experiment has been done. 1856 to 1863. 7 years he have worked with this experimental plant. So why he have chosen this experimental plant? Reason. Children, be sure that reason is a 2-mark question for your examination. It's a very big, very important 2-mark questions for your board exams. So why he have chosen this experimental plant? The first one is it is a bisexual flower. It is a bisexual flower, so it can be easily emasculated. It is a bisexual flower, so it can be easily emasculated and we can convert them into a unisexual flower. So the second important condition is these are clistogamous flower. Since they are clistogamous flower, that means they can produce assured seed cells because they have closed floral appendages. Since they have closed floral appendages, they can produce assured seed sets even in the absence of pollinators. The third is short lifespan. Therefore, single cross can produce many seeds. So therefore, more is the sampling size, because we can have an adequate data for analysis, and he used mathematics to explain the concept of biology. So more sampling size. Okay, what is the another reason for choosing this plant means it requires only minimum space and time, and look about the availability, the available throughout the season. Okay, and in this Mendel worked with 7 pairs of contrasting traits. Therefore 7 into 2 is equal to 14 contrasting traits. So he worked with 14 contrasting traits. Well children, the last point that we have discussed, why did Gregor John Mendel chose an Parzium sativum is he took 7 pairs of contrasting character. This session is very important. Why? Because in this we are going to learn the dominant as well as the recessive trait taken for all the 7 contrasting characters. Along with that, how to assign the symbols and put the alleles for the controlling character we are going to discuss. So these are the 7 pairs of contrasting character taken by Gregor John Mendel. The first one is stem height, the second one is seed shape, the third one is the seed color, fourth one is the flower color, fifth one is flower position, sixth one is pod shape, and seventh one is a pod color. I'll tell you about all this. In this, for everything he saw some contrasting traits. Stem height, he saw that tall was dominant over dwarf. Okay, how he identified that tall is dominant, why dwarf is not dominant, how did he choose? By performing a test cross. I'll tell you what is a test cross. Then is seed shape, in that he saw that the seeds which were round were dominant, which were wrinkled or shrinked, we are going to call as wrinkled, it was recessive. Then in seed color, he saw that yellow was dominant over the green color. Next is, look up at the flower color, he saw that violet color was dominant over the white colored flower. Next is the flower position, he saw that the flowers with axial knob, they were dominant, and one with terminal knob was recessive. What is pod? You all know that if this is the pea plant and this is the placentation where the seeds are there, so this one is called as the pod. In the pod only the seeds are there. So pod shape, one was full and other pod, you look up, it was showing some wrinkles which was called as constricted form. So based on that, the pod shape full was dominant over the constricted one. Okay, fine. The pod color, when you take into consideration, here you see yellow was dominant over green in case of seed color. In case of pod color, green was dominant over yellow. Now how to assign the symbols for the allele? So always take only the dominant trait and use a capital letter for dominant one. Or for the factors, for a single factor, take only the capital letter only from the dominant trait. And for recessive, put a small letter. People don't use any other letter. For example, the alleles for tall will be capital T, comma, heterozygotes, capital T, small t. Dwarf, no DD, no SS. Opposite means capital letter for tall, and for dwarf letter you will put a small t. So allele it is small t, small t. Come with seed shape, round. So you have to put capital R, This is as per the rules of first law of dominance. Characters or factors always occur in pair. In dissimilar pair, one pair dominates the other. That is for heterozygotes. So round, so wrinkle means, don't say sir it should be R and small w. No. They are there in the homologous chromosome. And there are contrasting characters. Tall means short or dwarf. But don't put T and S. T and small t. So here it will be small r, small r. Now for seed color. So we have yellow, which is dominant for homozygous, capital Y, and for heterozygotes, capital Y and small y. So it's given green. I know most of the students they put G. It's wrong. You should put small y, small y. Next is for flower color. In your NCERT textbook, there is a mistake that with a test cross they have given W. That is really wrong. But in the NCERT book, it is given as a small w and capital W. Recent edition, they have changed, children. So we have capital V, small v, and for recessive trait, small v. Because heterozygous homozygous recessive can only reveal the recessive characters. Flower color, capital A, and small a. Here it is small a. Here it is capital F, small f. Here it is small f. Pod color, capital G, small g. So never try to use the recessive character for assessing the symbols. While assigning symbols, take only the dominant trait, put them in capital letter, and for recessive trait, put by a small caps. That is how we have to put here. So people, tall means don't put short like this TS, wrong, or dwarf TD, wrong. Yellow, green, it is wrong. Yellow, and this is for recessive trait, small y you have to put. So this is how you have to assign the symbols for all the 7 pairs of contrasting characters. Please watch my previous video and please watch this video also. Combining these two, the next class we are going to see something, it's very important, Mendel's monohybrid cross. I am going to teach you tomorrow, followed by the monohybrid cross. Even I am going to discuss with you all, not only monohybrid cross, the meaning with explanation of the 2 laws framed by monohybrid, law of dominance as well as law of segregation. So thank you very much, children.